Hello, OIS podcast audience. It's a pleasure to be speaking with you again. For those of you who don't know me, my name is Rothman. I am a clinically practicing ophthalmologist. I'm a glaucoma specialist by training, and I spend about 50% of my time taking care of glaucoma patients in the office. And the remainder of my time is spent running InFocus Capital Partners, which is an ophthalmology-focused venture capital fund. InFocus Fund 1 has been fully deployed. We actually had our first exit about 5 months ago now, and we are currently navigating the remainder of the portfolio, which is fortunately doing well and propagating through some very interesting times, which we will talk about in a few minutes. And we are contemplating the launch of a Fund 2, hopefully in the near future. As some of you may know, I do the OIS podcast because I think it is important to keep the spirit of entrepreneurship and innovation in ophthalmology moving forward as aggressively as possible. And I've had the privilege over the past few years of doing these to speak to some amazing innovators and leaders, and today is no exception. Having the privilege to speak with Rajan Shukla, who is the founder and CEO of Bright Star Therapeutics and Vital Tears. And hopefully he and I will make sure that the entire listening audience is aware that we need to continue to focus on the anterior segment, anterior ocular surface, dry eye, and lots of other things besides dry macular degeneration. So Raj, thanks for taking the time to be on the podcast today.
Absolutely. Thanks for having me today. Looking forward to it.
Raj, I think one of the things that's become customary for me in these podcasts is to have the people who I'm speaking to kind of describe their journey. You know, everybody wants to know how people end up in ophthalmology. There's almost always a unique story behind everybody's journey into this space. Not everybody starts out in ophthalmology, but once you're here, most people stay. I kind of call ophthalmology the Roach Motel of life sciences. Once you're in, you're never getting out. But why don't you tell us a little bit about your background, your history, your story, and how you ended up, kind of building Dry Eye Company?
Certainly. Well, my story is a little different. It actually starts before I'm born. So a number of generations, actually. So my grandfather comes to the U.S. in 1970. He was a special education teacher and moved to a small town in Kentucky, north of Lexington, called Cynthiana, about 15,000 population. And he's just a pure entrepreneur, though, in addition to being a special ed teacher. So he's importing hammers and nails. He had a brass business for a while. And then he had a neighbor named Dr. John Stevens, who was an ophthalmologist. And he went over to his house one day and saw some instrumentation and thought, I could probably get instruments like this and sell them. And he heard about, how much they sell for, and was even more interested. So we got some instruments, went to the AAO in probably late 70s, 1978, 1979. And he calls my dad, who was in college at the time, and he says, "Son, we're rich. I just made $13,000 in 5 days," which was probably more than his whole salary as a special ed teacher in 1979. He says, "We're quitting everything else. We're only doing ophthalmology." So out of his living room in Cynthiana, Kentucky, he starts a ophthalmic instrument company. My father joins in 1982. When he gets out of school, then they move to Lexington. At that point, around 1985, they start an IOL company. And they do a very small seed round, about $150,000 just from local ophthalmologists on the backs of, just trust us, we're going to make IOLs. So they buy 2 lathes in a mill. People trust them enough to give them the money for that, and then start an IOL company. And then a few years later, that's when I enter my ophthalmic career as a 10-year-old lathing milling IOLs at our factory here in Lexington, Kentucky. And I've been in ophthalmology ever since. And so, I think a number of your guests, as opposed to that, I didn't have any other interests or careers before this. So
You didn't have a choice, actually.
Exactly right. So I start when I finish school. We're a small company at the time, and I honestly, I'd be lying if I said at that point I knew anything about patients or even thought about them. I was a mathematician by training from school, and I came very much focused on processes and improvements. And I spent the beginning of my career working in a lot of different areas. And so kind of as a small business, everybody does everything. So I get exposure to manufacturing, sales, marketing, everything that there is. Um, Things start to change for me a few years into my tenure there. I do a project. This is the first outside project I did. I did a co-development of an iris expansion device called Expand with a gentleman named Ron Dykes from Diamatrix, and learned a lot about product development, manufacturing, what product launches look like. Um, but more importantly, I went into surgery for the first time. So I think I'm about to 25 years old at the time. And Ron Dykes takes me into surgery. We go into Dallas, and we've been working on this device for about a year. I made a lot of them kind of personally, those early runs. And so for the first time, I'd spent since a 10-year-old, I've been lathing milling IOLs. I'd never seen live an ophthalmic surgery. And so I kind of have this moment where this physician saves all of his complicated cases for 1 day a month. And so we're in there, we're using our iris expansion device, we're waiting for Flomax people to come up, or floppy iris for other reasons. And I just have this moment where the surgeon is operating on the patient, and I see this brunescent cataract, and then go completely clear from an IOL. And then it's just such a different moment. Like I just had this connection to the patients at this point. It completely changed the way I thought about work and the way I thought about what I was doing. And then the next case comes in, he gets to use the device that I made. And it's kind of this perfect connection between, patient, physician, something you made. And you really have to experience that in order to really understand what that feels like. And so that completely changed sort of the reasons, the whys behind work. And I knew that I wanted to continue to do this. And so I get the next opportunity. We start to look at anterior segment. I have a long history in cornea. There are connections with eye banks, but to start looking at dry eye disease and really notice that there's no non-surgical, non-systemic treatments for severe dry eye disease other than serum tears, which is in the preferred practice pattern. We had one approved RX medication, but there's so many patients at this point that have residual symptoms after use of the only RX treatments that we have. I thought between 1 million and 1.5 million patients were still heavily undertreated. Did a lot of research looking at kind of the serum tears work that was done by Dr. Geerling, Dr. Sabota throughout the 1980s, 90s. And then we got an opportunity to start a serum tears company, which became Vital Tears with the folks at Side Eye Bank. And this was around 2014, and really started getting from the literature that serum tears were highly efficacious, and then spoke with physicians, 40 or 50 corneal surgeons, and they all said, oh, these are great, but they're not available. So it just seemed like an excellent opportunity to start a serum tears company from the ground up. And so we started that in 2014, and kind of notably, other people had tried to scale this up nationally and failed, including publicly traded companies. And they were really trying to take serum tears and fit it inside of an existing business model, when really what needed to happen was we had to start a brand new business model and build Vital Tears from the ground up to make them universally accessible. We wanted it to be as easy as writing a prescription for physicians, and then as easy as getting 2 blood draws per year for each patient. And so started a company to do just that. I had another formative moment. Again, when I'm working on this in the early days, I get to go into a clinic with your OIS colleague, Paul Karpecki, here in Lexington, Kentucky. And Paul sees 50, 75 ocular surface disease patients per day. So getting in with him and really getting to understand what the daily life is like. And there's people that just have a much different lived experience than you and I. And these are patients that also had somewhat been ignored, I think, by a lot of the investments that were aware of a lot of the investing dollars were going at the time. And so I really kind of felt like that we needed to work hard for them, and we knew serum tears worked. And so we need to get this into the hands of every physician so that every patient in need can get it. So that was another kind of real formative moment that just kind of made me realize that ocular surface disease, especially, was just heavily undertreated. Spent the last 10 years building, scaling up Vital Tears. We started with 3 physicians in Kansas City. Today, we've got more than 3,000 providers. We've this year passed over 100,000 serum tears orders. So we're kind of well along our way in our mission to make these universally accessible. Along the way, though, around 2020, I get a call from an OTC director at University of Minnesota, a gentleman named Andrew Morrow, and I worked on Vital Tears with him in the early days. And he says, "Hey, I've got this new technology that's being presented by a corneal physician next week. And I'd love for you to come up and take a look at this. I think it'd be really interesting." And they do these presentations to venture committees so they can get feedback on the idea en route to commercialization. So I came up to visit them and saw a presentation by Dr. Josh Ho at University of Minnesota, and he was presenting on a new version of CLET that addressed cultivated limbal epithelial transplantation, that addresses limbal stem cell deficiency, which I identified before. This is a disease that another anterior segment disease with terrible quality of life for these patients. You're in pain, you have low vision, or you're blind, and there's really just nothing available. And there's hardly any physicians or hardly anybody actually working on this disease. And serum tears worked up to a certain point, but beyond that, it really wasn't enough. So I knew that this was a huge untreated patient population. I was very excited to get to work on this. And in that meeting, what I realized immediately was we've got a brilliant physician, scientist, inventor in Dr. Josh Ho, and a fantastic research team there at University of Minnesota to be able to bring this to light. And that this was an important problem to work on, and that I wanted to in-license it and start a company for it. So I talked to the director of OTC there, and kind of in that meeting said I want to in-license. He asked me what I thought, and I said, "I think this is a phenomenal idea. I want to in-license it, start a company." And we did that. We did a year of preclinical research and really kind of were able to prove all the things we thought about the product in terms of how it can maintain limbal stem cells long term. And then we raised a Series A round in 2023, soft launched the product, and the national launch in 2024. And over the last 15 months, it's been scaling very rapidly. So that is my journey into ophthalmology.
Wow. So let me go backwards a little bit just to clarify for listeners. So what happened to the IOL company?
Oh, IOL company is still operating here in Lexington, Kentucky. We ended up, along the way became a heavy player in China. In fact, at some point, we were the largest international company marketing lenses in China. And so we ended up doing...
What's the name of that company?
US IOL.
Oh, okay. So you guys were US IOL. Great.
Exactly.
So, right. So you sold that company. Is that correct? I mean,
we did a strategic partnership with a Chinese-based entity there. Really just kind of was an excellent fit for us. And so we still operate the manufacturing and that company here in Lexington, Kentucky.
Okay, great. So you're still in the IOL business, but that's a separate entity. And now Vital Tears, which is the serum tears, separate entity on its own, cruising along, building market share, expanding the prescriber base, making it a little bit easier for patients to get. So that's kind of self-propelled at this point as well.
It is. And, with Vital Tears, what we're really focused on is market access at this point. So we do have a number of insurance companies that cover serum tears. And like you would expect, the utilization of the product is a lot higher for patients that have insurance coverage for it. So that's key focus for us. I just brought a new VP market access on from Horizon Pharma, and we're in discussions with a number of major insurance companies now.
Got it. Okay. So Bright Star is really the one focusing on some of the newer technology here.
Absolutely.
Okay. So let's go into a little bit about the procedure and the technology that you've developed here, because I think it's important to differentiate what it is you're doing from currently available therapeutic options.
Absolutely. So what we saw with Bright Star initially, when I licensed the technology, we were focused on a new version of cultivated limbal epithelial transplantation. So this is when you take limbal stem cells, you grow them on a substrate, and you transplant them to the ocular surface. Now, for patients with limbal stem cell deficiency, that is, let's say 75% plus, you've lost enough limbal stem cells, you need a stem cell transplant in order to re-epithelialize a clear cornea. And the options you have now are using your own cells via living-related allografts or CLAU or keratolimbar allograft from cadaveric tissue, or you can take autologous stem cell transplants as well, taking biopsies. There's a couple people working on that as well. Ocular notably got the very first advanced medicine therapeutic approval for autologous limbal stem cell transplantation. The issues that we saw with this were that when you have a patient with severe disease, if you cultivate limbal epithelial stem cells on most substrates, amniotic membrane, a fibrin membrane. What happens when you transplant them is the membrane will break down, and then you get terminal differentiation of those limbal stem cells, kind of normal wound healing. When the epithelium is damaged or removed, your native limbal epithelial stem cells grow out of the limbal niche to repopulate the corneal epithelium. When they reach central cornea, they differentiate terminally. So they don't have that same proliferation potential that you get, and you end up with just stratified layer of mature epithelium. Then you get 6 to 9 months a lifespan on those cells. Now, with our membrane, what we found is that it has special proteins that are able to maintain the stem cell phenotype. So what you get is a potentially the first long-term durable version of CLET, where you're getting pseudo-limbal niche, so you're supporting the limbal stem cells long-term, and not just transplanting mature epithelium, but actually transplanting stem cells that will last long-term on the ocular surface.
So how does that happen? So can you describe a little bit about because I think, at least for the cornea audience that will listen to this, they get it, right? They know.
Right.
But for some of the other people, it's like, why is this just different? What's different about your technology compared to regular limbal stem cell transplantation? What is the special
Absolutely.
sauce that you're willing to discuss so that people understand the differentiated features of this?
What happens with our product, we're actually using a specially processed decellularized Decimase membrane allograft. So this is a membrane that when we look at the anterior side, you have these proteins that are basically left over from the in utero development of the eye, such as vitronectin, spark-beam 40 osteonectin, and they are proteins that are also found in the limbal niche. So the limbal niche is the only area of the eye where the limbal stem cells can live long term, and they need this protein signaling to be able to maintain their stem cell phenotype long term. So that's what's so special about it. That and it is optically clear. So when you transplant it, you can see right away, unlike opaque membranes like amniotic membrane. And then you also have high resistance to degradation. When you think about this, clinically, we see a decimetacel, where you've got basically a thin membrane between your eye and the outside world, and that can be stable for months to years. This is highly resistant to degradation. So you have this optically clear, long-term stable membrane that can support limbal stem cells. And so that's what can enable us to potentially provide the first long-term solution for these patients.
So does that mean there's no terminal differentiation of the epithelial cells because they're surrounded by this decibase membrane protein environment that allows them to maintain their health? Is that what happens?
It's really interesting when you think about it, that when you have the normal responses, that you get the cells, they differentiate into transient amplifying cells, and then they differentiate into stratified layers of mature epithelium. With our membrane, when you culture limbal stem cells on these membranes, you'll see normal stratified epithelium, but you get this special layer, the bottom layer of cells that touch the membrane, they maintain their stem cell phenotype. So we've seen this in vitro and gotten to statistical significance versus kind of your normal corneal environment as well as amniotic membrane. And so that's really what makes this so special.
Huh. So is there a procedure involved for the surgeon? How is your product prepared / supplied to physicians? What's the nature of for most transplanted tissues of the cornea, people get a corneal button and they have to prepare it and do things to it. And, now I think we're starting to get a little bit more of preparation from eye banks of desmetic membranes that can be planted for, let's say, DSO or other procedures where we don't need full thickness corneal buttons. But how does it work with your product?
Absolutely. So I'll first mention that the stem cell product I'm discussing is called Bright Stem. That is currently
Okay.
in development right now. So we are preclinical. We're going to be doing our IND enabling animal studies this year. That is the membrane plus cultivated epithelial limbal stem cells. It's actually pretty remarkable. We're able to create these out of one single donor cornea, and it creates this all-in-one construct that surgeons receive and then transplant to the ocular surface. Now, the product that we have available commercially now, we talked about this product for severe partial and total limbal stem cell deficiency. We have another product that we developed along the way called BrightMEM. And this is simply the membrane by itself. So we're kind of thinking you know, we see in the lab, this has these amazing properties to help maintain limbal epithelial stem cells. And then we started thinking about, now, how about patients that do have enough limbal stem cells to potentially repopulate without a stem cell transplantation, but have some bad epithelium, say some neovascularization, you've got conjunctivalization, and you have some of this whorl-like keratopathy. So what we did is the surgeon initially used just one small quadrant of this. He did a superficial keratectomy. This was before we launched the product, when it was in development. And he transplanted a quadrant of membrane onto the ocular surface after doing an SK on a patient with partial limbal stem cell deficiency. What he noticed is that he got reconjunctivalization in the 3/4, and then the single quadrant of where we transplanted membrane remained clear long term. This was kind of a remarkable moment. And so what we theorized at that point was that normal wound healing, limbal stem cells are growing out of the niche to repopulate. And then instead of just terminally differentiating to repopulate the epithelium, they are seeding on this membrane on the ocular surface. And so we can use this for patients to prevent or push out the need for limbal stem cell transplants. We've got a aniridia study that's going to be publishing, a study in analytics that's going to be publishing later this year. And then we have launched this product in April of 2024 and been scaling it up rapidly, with kind of the two key indications being that visual rehabilitation in limbal stem cell deficiency, but also persistent epithelial defects. And so imagine that you have a patient, 7-10 days out, maybe you've done one amnion, you're still not healing the ocular surface. The procedure works by debriding out wider than the epithelial defect, transplanting our membrane on, gluing it down, and then it reepithelializes over that. We've been seeing really fantastic results from this, and we'll be sharing some top-line data shortly.
So is that a non-surgical procedure? Is it really just, you have to just get the membrane to the right size, your membrane to the right size to cover an area larger than the defect, and then fibrin glue or something like that in place? Is that how it works?
That's exactly how it works. Now, it is done in the ASC, but that's largely around billing because this is a corneal transplant.
Right. Wow. So that's fantastic. So people can buy BrightMEM and basically just have to cut the right size piece. Is it something they do with a tree find? Is it hand cut? How does it? Can you just give me like a brief overview of the procedure just so people understand? Because it sounds fascinating to me. I mean, as a glaucoma specialist, I'm a wrecker of corneas, right? My job is to destroy the cornea with glaucoma surgery or topical medications, and then have somebody else in the corneal world try and fix it later on. But it sounds to me like this is something that would solve a lot of the problems that I'm probably partially responsible for.
Yeah, absolutely. And the way this works is that it comes as a whole cornea with the BrightMEM already peeled, and then you debride out about a 0.5 millimeter wider than the target BrightMEM size. You take the whole, it comes on a scleral rim, comes like as a whole cornea, as you'd imagine, goes to a tree find block, a suction block, you punch it out to the desired size, and then then after you get the surface very dry, you transplant that button, let it dry for about 2 minutes, and then you just slide the Strobel button off. You have the BrightMEM down, use some BSS to smooth it out, and then fibrin glue to seal the edges, bandage contact lens, and then that's it. So it's a very fast procedure.
Wow. And how long does the bandage lens stay in place?
We leave that on for 1 month. And one of the reasons here, while developing this, I have a product development system that I've developed called COMIS, and use that with any product. It's called Comprehensive Outcomes Management Improvement System. And it's a process by which you collect clinical data and refine the outcome in a structured and anecdotal way. And then you improve the product, the procedure, as well as the surgical training, and then physician-facing materials. So kind of what we found out is that oftentimes you could be reepithelialized sooner than a month, but it's best to leave it on if you have someone that's a little slower, because a lot of these patients have impaired healing, that if you take it off before it's reepithelialized, it can put a little bit of wrinkling on that graft just from the suction, since it's very thin. So we leave that down for 1 month. But kind of what patients find so remarkable, one of these things is that typically they're pain-free after they've had the procedure. And that is not the case typically prior to if they have a painful epithelial defect. And then also, unlike amniotic membranes and other products like this, you can see right away because since it's optically clear. So people kind of know, oh, wow, I wasn't expecting to be able to see and feel as good as I do right afterward.
So there's a little bit of procedure, a little bit of a learning curve. It does take a little bit of effort. It's done in an ASC, but there's no suturing, there's no cutting. There's kind of just really about making sure you get proper placement of the membrane on the right spot and
Absolutely.
and smoothed out. Huh. Okay. Well, that's pretty interesting. Um, and you're currently selling this product.
We are absolutely. You can order directly through Bright Star, and we have distribution across the entire United States now. And we've got more than 100 surgeons now post-launch performing these, and rapidly growing every month.
So now let me ask you a question. So it sounds to me, I mean, just from the understanding that I think I have of what you've created, there's got to be other applications for this outside the cornea.
Yeah, absolutely. And when we think about the membrane, we talked about our pipeline Bright Stem, which is the membrane plus limbal stem cells. We also have Bright Ret in development as well, which is that membrane, and it really does a good impersonation of Brux, where you've got nutrient
Okay.
transport through and kind of some proteins. Potentially, we have some anti-angiogenic proteins as well. So we're looking at constructs of retinal pigment epithelial cells, cone progenitor cells, and our membrane as well.
So interesting. So you're going to pursue a retinal indication for this. That makes
Exactly.
kind of sense. Yeah, that makes kind of sense. Well, and we can talk about this for a minute, but I think one of the reasons why I was excited to kind of hear what you're up to is because the last few years have been difficult for everybody, at least in the venture side of the investment world, but certainly for a life science venture. And in ophthalmology, I think, we've taken a particularly big hit just because of the, paucity of outsized returns for investors that are looking for outsized returns in down markets, right? And I think that a lot of that has led to somewhat of a, let's say, oversized focus on retinal disease. Everybody wants to find something for dry AMD or diabetic maculopathy or whatever. And, I think that we've seen this kind of decrease in the funding cycle that's allowed, certain disease spaces in ophthalmology, dry eye, glaucoma, whatnot, kind of slow down a little bit. Right. And would you agree with that? Would you agree that there's been a little bit of a defocus on, the unmet need of everything outside of retinal disease?
Absolutely. It does look like that's kind of where, both from a public markets as well as from a venture dollars perspective, most of the investment is going. And it is, I mean, I completely agree with you that it is not where we need to be long term. And I believe strongly that if you find a product, if you're able to develop products that work for patients that have significant disease, low quality of life, and large enough patient sizes, that you will have financial success on that. And so people shouldn't be as scared of that. I mean, we've seen some successes. Oxervate obviously has been a significant success. I think some of the other ARCS medications that are with for dry eye disease have had success as well. And so people don't need to be as scared of it as they are. I think there's fantastic returns potential alongside of this.
Well, then here's, your company, you're an example, right? Here's an example of somebody who's built out a growing business in dry eye and corneal disease during a time when there's been, very little funding activity in dry eye. And yet I think the unmet need in ophthalmology in areas, for example, like ocular surface disease and dry eye is pretty significant. We have, you know, not a lot of really great pharmaceutical options. I know that we've been reliant on cyclosporin in one formulation or another to really kind of be our go-to, bigger gun for treating dry eye. And I think that there's room for newer technology and advancements. You're, let's say, at the far end of that spectrum where, we're getting to needing something surgical, but yet, there's room for all of these new technologies, and they're all going to end up needing to be found at some point because we're just not doing a great job of taking care of people with serious ocular surface disease.
Yep, that's completely true. And it's really about anywhere that you have patients with significant symptoms. I think kind of part of the hard part about this is, dry eye trials ocular surface trials are notoriously difficult. It's hard to recruit for PED trials. It's hard to show efficacy consistently when you've got this, pretty heterogeneous population of folks. And that is where you really need to be thinking about how do we dial in giving a first name, last name to the disease of these patients. And so kind of better diagnostics, and also thinking about, for the longest time, we talked about aqueous deficient dry eye disease, evaporative disease as two distinct entities when most patients are mixed. You talk about there's so much focus on inflammation. When, from our perspective, and especially when we kind of think about from a vital tears perspective, from a serum tears perspective, that abnormal innervation in a patient. Now, this gets most discussed when we talk about neurotrophic keratitis, but it gets ignored when you really think about the broader dry population. I mean, a lot of these studies now are showing abnormal innervation, patients showing both higher than normal and lower than normal sensation. So those high sensation patients, abnormal confocals indicating neuropathic corneal pain. And that's really an area where serum tears is the standard of care along with Lotemax. Pedram Hammer famously, published that protocol, and largely that's kind of what people follow there. And then with neurotrophic keratitis as well, you've got, again, abnormal innervation of the cornea. And so ignoring that and using therapeutics that don't address it, that is one of the key reasons, I believe, why we have such a highly untreated patient population in dry eye disease. And I think that's why serum tears does so well. We have actually a PRO paper that top line will be coming out shortly as well. We're submitting for publication here this will be the largest serum tears study ever done. It's more than 1,000 patients. And we are showing response in a very high number of patients. And I think that for us, largely, that is because of this issue that comes with abnormal innervation. And, some of the biochemical components and growth factors that we have are really needed in order to address these patients.
Well, I think it'll be great when that paper comes out. I think a lot of people who are involved or at least interested in dry eye space will be looking forward to seeing what your data shows. But I think clinically that obviously is supported. Half the referrals that I make to cornea specialists, the patients end up coming back on serum tears.
Absolutely.
That's kind of what happens. And I think that ultimately the improved availability through, vital tears will hopefully make that easier for them. I've had patients who've had to, buy their own centrifuges and do all these weird things and to themselves in order to be able to get them. And it'll be great to see, I think, a broader availability through your company. Where are you guys in the funding cycle? Are you guys self-sustaining at this point? You're going to raise capital? What's your plan for the future?
Yeah, Vital Tears right now, we're profitable and continuing we basically invest all of the money that we make back into the company for largely focused around patient access. This is a similar story to the USIOL founding where we started this company with $180,000 in invested capital. And then, today we've, I think, done significantly better than you would expect based on that funding.
And what about Bright Star?
Bright Star raised a Series A round 2023. We are basically to break even now just on our tissue product. And so we are now pivoting to largely focusing on a continued ramp of that product along with the advancing the development of our cell therapy assets. So we're probably looking at we're most likely looking at around in the next 2 years.
That's be fantastic. And then obviously you'll need to raise more significant dollars, obviously, if you're going to move into the retinal space to do work for that. That'll be a bigger project for you.
Yep, definitely.
Raj, this is great. I mean, I really appreciate you taking the time. It's exciting, again, for me as both an ophthalmologist and an investor to, see that the spirit of treatment of dry eye is still alive and well, and that there are companies out there that are propagating this. I know that there's, lots of companies in development that are going to be looking for capital to try and produce products. You guys are out there. You've got things that people can, currently utilize to help their dry eye patients. And it's great to know that you're continuing to remain focused on, treatment of dry eye at multiple levels of severity. And I applaud the work that you've done so far in getting yourselves to this position. So thank you for that. So Raj Shukla, founder and CEO of Bright Star Therapeutics and Vital Tears, thanks again for taking the time. Look forward to seeing the results of your paper when it comes out, and hopefully we'll be back here in the future when you've got additional products to discuss.
was a pleasure. Thanks again, Rob.
All right. Thanks, OIS podcast audience. Looking forward to the next time we'll be speaking together.